Hi, everyone. I have a tough job first after Stefan. Stefan, nice to meet you. This was the first time I see you. But such a great continuation to go and see how the Obe-cel went from the university hospital all the way to the patients becoming commercial. I want to start first to say that this year we celebrated our 10th anniversary. And what a great reward will be if we can gain the licensure from FDA at the end of the year. So, yes, I already said that we are still in the stage of our review with the health authorities. It is a lot of fun, I have to tell you. This is probably one of my favorite parts, going back and forth with the information requests. So what I'm going to go today through, I'll go to the journey, how the whole process started from our founder, Martin Pule, amazing person to work with. And our Obe-cel had 6 years journey from UCL all the way to our new facility, Nucleus, which is not too far from London. And we built it for a year and a half. It is really great achievement for the whole company. I will go through the process control strategy establishment. This is something that it is not so exciting when I talk about that, but it is so relevant for where we all want to go. And I think Miltenyi wants to go in 2 years there. So hopefully this journey may be helpful. And of course, I'll finish with the commercial process readiness and how we did approach our comparability studies and our PPQ studies. So Obe-cel, and it is a CD-targeted or directed genetically modified T cells which are targeting the ALL and in adults. So it started in UCL, as I said, and the first process was manual open process in some of the cases. The patient, the leukapheresis material was incubated with the Dynabits. As we know, this was the first process for the CAR T cells. We did the whole transduction with the lentivirus of the patients, PBMCs, there was no selection. And the Dynabit removal was, of course, challenging with all the proofs that have to happen afterwards. So this was very acknowledged that we have to move from this way of processing towards something that is more reliable, and this is when the Prodigy platform came in place. And I love the colors, by the way. And very sleek platform which answers a lot of questions and eases the manufacturing and the development of the process. So with the implementation of this still in the hospitals, I have to mention that we're still in the hospitals, and this is still where we are manufacturing our for process B clinical trials, I may say. But the implementation of the Prodigy platform was already there. Moving away from the hospital settings, we are going to the cell and gene therapy hubs, which was catapult for Autolus, and it is again in Stevenage. In this catapult facility, we had the clean room, and we were able to establish our first going to pivotal pivotal phase of Obe-cel, we will establish our process there. Along with the process development and the continuous process development, I may say, we did future automation steps, and we started with our process characterization. I will pay more attention after that on this very step of the process development. And we did the process control strategy establishment, where all the critical process parameters, the in-process controls need to be put in place. Very important for the longevity and for of the drug, and at the same time, all of us want to have controls and manufacture consistently the drug for the patients without going out of spec. I mean, for the clinical trials, it's something that, okay, we can afford, we can go back and forth. For commercial, it is something that developers have to take this under consideration and ensure that this is not going to happen, or it is minimized out of specs. And we did the comparability studies and the PPQ studies in nucleus. Very nice facility. So I'm not going to go through this picture. I simply wanted to have the picture how we started, and everything was so, bulky and difficult to control. But this is when we went to the Prodigy system. And as I said, our goal is to be consistent and reliable manufacturing for the CAR-Ts. And we are targeting in our nucleus facility to manufacture more than 2,000 batches per year. So it is something that we're looking for, having a process that is robust to be able to meet the patient needs. So the next slide, it is a very trivial, I may say, how the CAR-T cell manufacturing process goes. But what I want to say here is during the process development, and specifically during the pivotal stage of the process, we started the characterization of each step of the product at each step. It is important it was very important for us to do this in order to establish the proven acceptable range of the process, where the process can operate very robustly, and also to get which parameters are critical, which parameters are non-critical, and how we can control the process. So going to this slide probably is a summary of everything that we're going through as companies, from the clinical development and process validation stages, and how the clinical development different phases are linked to the process design stages. So for Obe-cel, when we started in the beginning, we relied, of course, on some of our historical data in the beginning of the phase 1, and we continued to gain the knowledge through the process development. Very important part at this stage is to do the initial assessment of the presumptive critical process parameters. And of course, this was and still is a live document which goes all the time. We go and review the document. We do the assessment, reassessment, and about the process characterization. Yeah, probably this is not the most fun of the work, but this is the most important part of for us to understand where the process actually is very controlled and how we can do that. And for this to be accomplished, we needed a very robust scale-down model. And if you're working towards this, some of you are at this stage of your drug development, and if you don't have such scale-down model, probably it is very good to do so. and to make it. It is going to help a lot. Because of that, we did 250 small-scale runs. So you cannot do this with the Prodigy, right? So it is important. The other thing that is specific for the Obe-cel and why the process robustness is very important to be established in the beginning was the specificity of the disease. We deal with a lot of different initial material of the patient. Some of the patients, about 15% of the patients have less than 5% of the disease burden during the pre-screening. Around 55% have disease burden between 5 and 75%. But another 30% of the patients are really highly disease burden with highly diseased material. And this eventually makes it very difficult for a fit-to-all process. So when we developed the process, we had to take under consideration our variability of this material and how we can ensure that in the future, even the patients with the high disease burden can receive the dose. Obe-cel is also it is a split dose regimen, so it has, and depending on the disease burden, the decision which dose to be infused when is made. So it is, for us, it's important to know these details of our patients. Moving away from this remark, the process characterization is done in the preparation of the process, moving forward with the process control strategy, drafting the process control strategy was a great target for us. And we're going back to this journey. It was a lot of work for the whole company, but it was very rewarding. I have to tell you, when you see everything in one page with all the controls, it's amazing. When we move to the stage 2 of the process validation, this is when the process qualification needs to take place, and this is when pretty much we can talk about the readiness of filing and going to FDA. So we did the PPQs and the comparability studies, and I'll finish with that after my next slide, which is, I'm not going to go to this slide, but I simply wanted to be as messy as possible, just to show you that the interactions between the different functions in the company is extremely important, and the earlier it starts, the better. Because as cell process developers, we depend on every single role, and also they depend on our knowledge. So we need to constantly feed back and forth, and this loop needs to be refreshed almost daily. When I was in my young age, I was thinking, oh, I can do everything, right? No. Now it is, in order something to get good products, right? I mean, we have to work together very earlier. And this is usually my message when I start with my new people. I just try to find the right people to get the message, to get the right information, and to be able to do the things right from the first time. Yes, this is I have it in my office, this slide, along with all the data. Of course, I mean, unfortunately, I cannot show you all the numbers, but I can show you at least the strategy behind that. Where Miltenyi, right? If you look at the material controls, we have a lot of your reagents here. So the process controls are linked to each process steps, and how we define to each process steps, what controls need to be in place, what needs to be monitored, what needs to be controlled, what is critical, what needs to be defined. It comes after our experience was put together in a data from the clinical trials, from our characterization studies, and also from our tech transfer studies, tech transfer runs. Of course, this slide represents also the analytical testing where we would do. I'm not going to go to this very difficult task, I may say. Analytics are very important for the understanding of our product. And if the analytical, and one thing I want to mention here specifically for the for the CAR T cells, when the analytics are made, we have fantastic analytical colleagues, but sometimes they're seeing analytics in a very distant way, right? They don't have the material is very clean, they clean it for them to process. But what is important, I found that when the analytics are very closely related to the process steps, this is when the right outcome comes, and this is when the controls are much better. You know, I think it is something that I have learned in hard way. Yeah, and the most important part are the CQAs from these process controls and our release testing. The tough questions, where people change facilities, we change facilities, and what we need to do to evaluate the comparability. So we did change our facility from catapult to nucleus. We did the impact assessment of the critical quality attributes and parameters that might be impacted by the change. So this is something that you cannot go through the whole list, right? It is a laundry list of many parameters. So what might be impacted of the change needs to be assessed by the scientists and by the people who are doing the process, and based on that, the CQAs are going to be defined, and after that, we did our statistical evaluation approach, and we defined our comparability acceptance criteria. Of course, the execution of the studies, and after that, the results. So this is a nice slide that was made in 2015 for FDA draft guidelines, and how the suggestions about the equivalency needs to be taken when we change processes, when we change facilities, when we change everything that might impact the product quality. And they said we recommend the CAR-T comparability be assessed by side-by-side testing using the same cellular material when possible. Of course, it is difficult for our product to go and get apheresis material from patients and split it, but we did a lot of work with the healthy donor material. And of course, we needed to prove that this is a representative model today in order to do that, but we ended up with a very nice protocol. The execution was flawless, and our comparison between the two sites was accepted. So it is really nice. Spend time, if you enter into this level, spend time to do nice protocol, talk to the agency back and forth. They're good. I like the agencies, I don't know. Actually, it is very helpful to move faster. And probably something that you may ask, okay, what did you do? So I had this is the last slide here. For me today, we did for the comparability and the PPQ studies, we did the integrated approach, where actually we use 5 split apheresis runs. And we split them into the 2 facilities, and we run, of course, one of the protocol was for our comparability studies, and the other one was for our process performance qualification studies. Both of them require different data generation, but we were able to separate these, and at the end the reports ended up very good, and we got the data that we are happy to report to the agency, and moving forward with them towards our review, and hopefully we can get soon the approval. So this was my going through the journey of Obe-cel, and specifically the processes, the controls, and the involvement of the process, the thinking. Thank you very much.
No? Yes. Okay, it's easier like that. Thank you very much. It was really like an incredible journey. We are open for questions from the audience. Anyone? We don't have so much time for questions, but maybe one or two. Okay, Silvio.
Hi, Cookie. Very insightful presentation. Really liked it. I have a question regarding the process scale-down model. So I think the device is quite particular, the Prodigy. In which unit operations did you see good transferability between the scale-down model and the Prodigy, and in which it was really challenging to achieve that? Could you maybe comment on that?
I think the expansion is quite okay. The expansion stage is comparable. I think in terms of where we operate with our range of cells and transduction, I think the transduction was also comparable. The most difficult part is at the end, right? When you have to, if we have to freeze, if we have to do some stability. This is not something that I would suggest the scale-down model to be used. But for the selection, I think it is comparable. Activation as well. I think it's taking under consideration the geometry and at the same time the kinetics as well. When you do this scale-down model, is helpful to get as close as possible. But I found that what we have done was pretty suitable for our characterization studies.
Ulf, you had a question? Can you go to the microphone?
No.
There.
You can understand me?
Yes, yeah, but we have people online that don't hear you.
At the beginning of your talk, you mentioned you did categorize your patients in three groups: low, middle, and high tumor burden.
Yeah.
Did it result in what was the result and what was the outcome? Are you dosing differently? Are you?
Yes. So for the dosing, I mentioned that we have a split dose, and if we have a high disease burden patient, we actually start with the lower dose, and after that the second infusion is the high dose. And if the patient is the other way, we do start with different dose, and after that add the rest of the dose later. So it does play a huge role in the safety profile of the drug. I think that this is where you are going, and I do believe that actually this split dose regimen is giving the Obe-cel a huge edge.
Okay.
There's another one.
No.
I can, but I'm not sure.
I'm a professor in pediatric rheumatology based at UCL. So it was very nice to see the story of starting within an academic environment, UCL, and then obviously the spin-off company. So can I take you back the other way around, how open are you as a company to new academic ideas and proposals, if that comes from an idea that perhaps does not seem to be your top commercial priority?
I work with Martin Pule, right? It says it all. No, I think in Autolus we are actually very open to hear everything that is going to be potentially beneficial for the patients. Martin is a fantastic person who does take this under consideration. Of course, we have to find the balance, but I think in the early development, how the current model is for Autolus, is very possible to start with something in the university, even if it doesn't fit the probably. I cannot speak from the corporate level, right? Don't get me wrong, but everything I have seen is, I have not seen anything that is shut down on the business because of the business. So, yeah, if you have something